Hi, everybody. So my name is Thierry Bastogne. I'm a professor at University of Lorraine in data science for Earth, but I also Chief Scientific Officer at Cyber Nano. And first of all, I thank you, the SGS team and Philippe Grimm, to invite me for this presentation. And my presentation will be mainly focused on quality by design. And so I just start to introduce quality by design. Let me just remind you all the steps, classical steps of drug development. And just below, you can see the TRL scale. This is a technological readiness level, so a kind of scale to measure the maturity of innovation. And below and at the top, you can see the quality by design. And we can define quality by design as a kind of good development practice. And the idea is to go as soon as possible from the level 2, so just after the discovery, up to level 7, that is at the end of clinical phase 2. And the official text for pharmaceutical quality by design is the guidance ICH-Q8 here, strongly recommended by FDA and EMA to develop new drugs. And they define QBD as the best practice to provide all the information required to fit in the module 3 of the CTD documents for any marketing authorization. In other words, QBD can be defined as a practice to optimize CMC studies. Another aspect of QBD, often we talk about also quality by data, because statistics are really omnipresent in the quality by design process. So that was really to sum up the quality by design and what could be now the benefits of QBD approximately 15 years after the publication of the ICH-Q8. Finally, two studies converge to conclude that we can reduce time and cost of the development between 20 and 30% with QBD. The first study is an economical study. The second is more recent, and it is more an industrial demonstration about how the QBD help the authors to really reduce the time of development. So we know that QBD works, and this is not the only papers, but now, 2 years ago, another paper confirmed that QBD is becoming really the standard of development, but Europe is very late compared to the implementation of QBD in US. And we have identified 3 reasons for that. First of all, QBD is not a magic formula. There is a large bad understanding of all the components of QBD. And ICH-Q8 is a guidance, not a method. And that could be a first barrier. Now we also need a large skills in biostatistics because we need efficient tools in statistics, and it could be another aspect, another problem. And this is why we need also a kind of pluridisciplinary support and pluridisciplinary teams in statistics, in biology, of course, biotechnology, and that's another barrier to facilitate the implementation of QBD in practice. Now, what we have developed, the first objective was to address the 3 limitations I presented just before. So we have worked a lot on improving the methodology associated with QBD. A second compound was to improve and develop new statistical tools better appropriate for the implementation of the QBD. And the last aspect was to build a pluridisciplinary team to facilitate and to better discuss with all the drug developers. So all the three ingredients have been implemented in what we call a SAS platform, SAS meaning software as a service platform. This is a technological innovation. A SaaS platform is a kind of software, but it is more than a software. How does it work? So imagine you are a biotech, you wish to develop a new drug, and you wish to implement the QBD to optimize the development and the CMC dossier. So you can connect on the platform. So you have a login and a password. And so step by step, in like a software, you are guided to progress in the QBD implementation. But when I said it is more than a software, it is because you are assisted by different experts, like for instance, a biostatistician, to help you to design correctly your experiments, to analyze your data, and to interpret the results. And to do that, the biostatistician, is using different softwares, different algorithms we are developing, like for instance, the Bayesian techniques, and once again, to integrate all the different ingredients to facilitate and optimize the QBD implementation. The first thing, when you log in, the first thing you will see is this kind of roadmap. Let me take a few seconds to present you the roadmap. This is kind of subway map. You can see main stations, the TRL level, so the TRL2, TRL3, the first proof of concept in vitro, the second proof of concept in vivo for the safety and efficacy. When you've got a drug developed in GMP, tested in GLP, you are at level 5, and level 6, 7, and 8 correspond to the end of phase 1, clinical phase 1, clinical phase 2, and also clinical phase 3. Okay, so when we are in this map, you can see some kind of small stations corresponding to key questions during the development of your drug. So it can correspond to different sections of the CMC dossier. And sometimes when we have not enough data to clearly answer to a challenging question, we can do a QBD analysis. All the QBD analyses are plotted by a kind of circle, but this is not obligation to do QBD after QBD. If you estimate that you have enough information to answer different questions, this is not necessary to implement a QBD analysis. So you just implement, apply quality by design when you have to address a specific key and challenging question in terms of development. And I would like to give you an example. So imagine you are there, and just to explain you the contents of a QBD analysis there. So you can see here, so the cycle corresponding to this part. So each QBD analysis is divided and split up into 4 steps. The QTPP, so the quality target product profile, in which you define the type of drug you want to develop and the key question you want to address. The second step corresponds to what we call the input-output modeling. It is just there to identify the key variables of the problem. After that, this is a design of experiments, like Eli presented just before. We often use design of experiment in biology. And then the second is the data analysis to finally answer to the initial question. So now we can give you an example. This is how we are working for a biotech in Belgium, Eterna, developing a nano carrier for the mRNA transfection. And at the beginning, the initial question at a specific step of development was how to find the optimal formulation of the nano carrier. And that was a key question. And at the input-output modeling step, we have identified 3 inputs, what we call the critical material attributes, so the composition or the proportion of 3 different lipids. And the critical quality attribute is the immune response, or more technically, the transfection rate. And we have a requirement, and the specification is to get a transfection rate greater than 50%. But this is clearly the objective. But we need to determine the relationship between the input and the output. To do that, we need to get data, and to collect the data, we need to design some experiments. So this is a specific Rockmore's hybrid design in which we have implemented each red dot corresponds to a specific experiment. We have only 11 experiments carried out in triplicates in vivo context. And after the experimentation phase, we collect the data and we analyze the data to get this. This cube, in fact, corresponds to the universe of all the possibilities of lipids. And you can see a green part, a green region. This is what the FDA and ICH calls the design space. In this part, in this region, we have more than 90% of chance to meet the requirement. So all the different points, green points, corresponds to different proportions of lipids with a maximum of chance to finally get the requirement we need. And yes, we have to meet. So this is an example. And after the analysis, so we know we have the design space, we can go to the next step of development. So this is a principle of what we have developed. So if now what is the different items we have to remember? The quality by design is really now a good development practice in which we try to estimate the risk in terms of efficacy and safety requirements predicted from data and data collected during experiments that could be in vitro experiments, in vivo experiments, directly applied on the product to be developed. The pharmaceutical QBD is an efficient approach of drug development if and only if all the conditions in terms of methodology, in terms of statistical tools, in terms of expertise are met. Otherwise, QBD is not a guarantee to do the things correctly. QBD must be implemented step by step. This is really important. This is why I presented you just before the roadmap. So we have different questions, and sequence by sequence, we address questions to progress surely step by step. The objective is to avoid to go back and finally to waste a lot of time. And at each step of development, QBD must provide relevant data and relevant information to efficiently complete the CMC dossier. As I told you in introduction, in fact, QBD is a kind of good practice to optimize the CMC dossier filling. Now, I just also add another point. It's now since July 2022, there is another QBD. This is the AQBD. The same approach, quality by design, but to develop robust and accurate analytical methods. And this is also something that will be more and more important in the next years.